It is my pleasure to record this presentation that I've recently presented at the European Society of Radiopharmaceutical Research in Coimbra, Portugal. It was a beautiful visit to a beautiful country, and it was quite an interesting presentation, and I think it sparked also a lot of discussion. So it is then my pleasure to also upload it on my YouTube channel and make it more freely available and hopefully have some more interaction on the topic. Just as a small introduction, why we are doing alpha emitters, because lutetium and beta minus emitters have gone quite far and progressed greatly, but we still can fine-tune targeted radiotherapy and also use or harness the more powerful effects of alpha emitters because as you might know, alpha emitters cause double strand breakage. It has high linear energy transfer in a shorter range, and it's less sensitive towards maybe radiation resistant and hypoxia and all of those things that can normally maybe cause the other therapies to fail at some point or not have that great of a benefit. And it can also have indirect effects like the bystander effect, activation of the immune system, and also abscopal effects. So when I want to discuss alpha emitters, I first kind of started to think what is the ideal? What would be the ideal alpha emitter for targeted alpha therapy if we could have everything we want, which is of course not the case. And I thought of some factors here, but if you have some other things you think we should also consider, please type in the comments. Actually, for the whole presentation, please just type away in the comments, or if you have questions, go ahead. It's quite a fun interactive topic. So nice of terbium to provide us with a lot of options, isotopic options, where the isotope behaves chemically and biologically the same, but it has different emission characteristics. So that is quite interesting. And if you could build your own alpha emitter, this would have been quite something I would add to my list. It should have stable chemistry, not only for during decay, but also just in general stable chemistry. It should be available, scalable production. It should also provide you with maybe one alpha emission. I think this is up for debate because a lot of people really prefer the in vivo generators to give you higher linear energy transfer in one go, in one shot, and therefore maybe harnessing a lot of extra power. It should also be directly quantifiable and safe to work with. And yesterday I was at a meeting Ismerat in Ghent in Belgium, and I heard another list of things that we should have a look at. So I'm going to mention them. I didn't add them here, but it should be achievable, applicable, adoptable, appropriate, and available. So the person that presented there said we should look at 5 A's. That's quite a nice way to say it as well. It's not maybe so focused on the chemistry and the radiopharmacy aspect as my list is, but we should actually look at that too. With achievable, it means good radiochemistry, synthesis that is developed. Applicable is easy to expand to other applications or indications. Adoptable means it should be allowing you to kind of work in the framework of the regulatory environment, and in safety and efficacy is for appropriateness. So yeah, and available, of course, I think we all have available or upscalable production, because I think that is currently the question that we have to face. That is the biggest. So for the progress so far, I think it's very interesting to realize that some alpha emitters have progressed quite far in the clinic, others not so much. And this could also be an indication of future usability of each alpha emitter. Actinium dotatate, as you can see, is already in phase 3 trials. Astatine throughout the years have been making progress in phase 1 trials, especially in Japan. Bismuth 213, there is some examples of first small pilot clinical studies. Bismuth 212, not so much. Late to 12 is gaining quite momentum. It hasn't been researched that much in the past, but suddenly there was quite a huge spark in interest. Radium chloride in the form of non-targeted radium for bone pain, palliation, and even treatments is still, I think, the most advanced alpha radionuclide usage. I was yesterday also in this meeting where it was suggested by the clinical side by an excellent presentation of Prof. Mike Satehe that radium-223 is still very much important because it can also be used in combination therapies with other radio pharmaceuticals, indeed even other alpha emitters. And then terbium-149 really is a little bit hampered by availability at the moment. Thorium-227 has been investigated quite substantially in the past, but it's not clear what the role of thorium-227 in targeted alpha therapy will be. So if you look at the periodic table, we have identified the hopeful 8 by this excellent review that you should have a read at. There is versatile chemistry, as you can see here. They are spread all over the periodic table, and I will be discussing this 8 radionuclides in alphabetical order, because I'm going to declare here now, I'm also the one that always says to people, I don't think it is wise at this point to have a favorite alpha emitter. I really feel very passionate about it, that we should give them all the necessary attention, because different institutions, different infrastructure, and different indications should naturally evolve into like a favorite alpha emitter, if I can say it that way. So if you think about PET radionuclides, there is, in my opinion, not a favorite one. I mean, fluorine 18 is the most used perhaps in some regions, but for this targeted approach, gallium 68 or fluorine 18 or carbon 11, all of them have their place, zirconium 89. So in my view, to so passionately argue for your favorite alpha emitter and discredit the others is a bit limited outlook. So I would prefer us to say maybe we give them all the necessary attention and see what comes out of it at the other end. It's a bit early also to make really passionate statements about a favorite. So I'm going to discuss them in alphabetical order. And if you have worked with this isotope or if you're curious about it, please type something in the comments. Have you worked with actinium-225 before? Or do you see yourself maybe very soon working with it? Or what is your interest in it? Or if you also have comments on anything I say and you might want to give a counter argument, please go ahead. So I will disclaim every time. I have worked with actinium-225 quite extensively. I was involved in the human applications in a phase 1 trial, PSMA based. And so I know this isotope quite well, this radionuclide. And yeah, I think I can have an opinion about it. So it is the most advanced clinically, of course. There is 27 molecules under development at the stage that this video is recorded, 24 registered trials on clinicaltrial.gov, and we even have an agent that has reached phase 3 stage. Actinium has a great production upscaling potential. There is quite multiple options and multiple people are solving the availability issue, and we will discuss that too. It's a radiometal, so mostly at this stage, what is in the clinic is database labeling, and there is quite a lot of difficulties with real-time quantifications due to the emissions of the actinium-225. So you cannot directly measure actinium-225 through alpha emission with most equipment. There is some research being done on that, but normally you would focus on francium-221 with a gamma emission of 218 keV, or bismuth-213 with a gamma emission of 440 keV. In order to measure accurately, you then have to wait for enough of the daughters to be produced and to be like in balance. So this is the current production methods for actinium-225 that is being investigated. Firstly, it is through the stockpile of thorium-229. It is 4M229 is sort of a limited research because it's looking at radioactive waste and it's gathering that. Yeah, it has its limitations, but it gives a really high specific activity product that is very pure because it's a generator-based system, and you can chemically separate very pure actinium-225 out of that, normally every 2 months. But there is a great article that I will refer you to that discuss all of these methods in depth. The other method is through the use of 4M232 by high proton energy bombardment and then chemical separation thereafter. This method makes actinium-225 contaminated with some actinium-227, depending on how you go through your production cycle. It can be optimized, of course. And then also the radium-225 isotopes that you can separate chemically out of your actinium-225. You will see that the radium-225 decays to actinium-225. So it is also being done that rather than using the bulk of the production like this with all of the actinium and radium isotopes, you would rather remove the radium isotopes out of the mixture, and then you can elute a very pure actinium-225 from that. However, you can only get about 10% of your yield than what you would do if you use this whole big batch. So you waste a little bit of actinium. Well, not a little bit, a lot. You get a very lower yield. The other option that's being actively pursued, especially in the industry, is the use of radium-226 in 18 MeV cyclotrons by either the proton route or the gamma route. Yesterday, I also sat in a discussion, well, at a presentation where this was discussed in great depth. It was actually a really good presentation. And there is, of course, all the problems of cooling the target and also dealing with radon gas that might be released. So this is a bit tricky and dangerous, but there is companies that is actively producing new technology that might make this quite a feasible route in the future. But you should be careful that your target is not overheated. Actinium-225 radiopharmaceutical stability is always a question. It gets asked a lot. So firstly, you have three mechanisms that can cause stability issues. Recoil energy, the chemical release of the daughters, and radiolysis. Recoil energy is the Newtonian principle where if you give a force of a high impact, you also have an opposite reaction, and it's often said that this great energy alpha emission will really remove the alpha emitter from out of the chelator system it is incorporated into. However, if you could get a chelator that is strong enough for the to accommodate this recoil energy and still keep your alpha emitter in there, it is also important to realize that there is also a chemical release of the daughter, because actinium-225 has quite a different chemistry from francium-221, for instance, that is the decay product in the system. So could you solve the chemical release? Of course, you still have to deal with the recoil energy. So it's important to know that both of these factors play a really important role. And then we also have radiolysis, and this is something that was a bit neglected in the past, but I'm glad to see some research being done on here. And it is really important to note that as with lutetium and other beta minus emitters, the high energy of these therapeutic radionuclides can cause transformation of the carrier molecules, especially if it's biological molecules, and this should also be taken into account. Should the energy that is released in the mixture change the conformation of some of the molecules, this can lead to quite a different pharmacokinetic behavior inside the body. You have the chelators, DTPA that was used in the past to have this interaction with actinium-225, but it was shown not to be the most stable option, especially in vivo. So currently DOTA is the gold standard, actinium DOTA, used for all molecules basically, but keep in mind that needs quite harsh conditions to label, heating at a higher temperature, and therefore a lot of effort has been done to develop new chelators, and the Macropa group is being investigated for more milder labeling conditions, and then also allowing the use of biological molecules for actinium-225 targeted alpha therapy. Macropa as it is now will not maybe solve the problems of the recoil energy and the chemical release of the daughters, but it will make it available that you can label biological molecules and might even assist with some of the radiolysis issues, because if you heat up and have harsh labeling conditions, of course, radiolysis and instability of the vector becomes even more important. The measurement of actinium-225 is always tricky. On this YouTube channel, there is another video that covers this, so I'm not going to do it in depth, but be aware that you have to wait around 2 hours or sometimes even a little bit less if you validate it to have an accurate measurement of actinium-225 quality control. So, any time you chemically separate actinium-225 by chromatography or by injecting in a biological system, you have to wait for an equilibrium to form. In other words, you have to wait that the daughter isotope is given off in the right amounts to create a balance so that you know you can measure accurately the amount of actinium-225. So we never measure actinium-225, but we use francium-221 as a surrogate for the amount of actinium-225 that is there. So when you first do your ITLC, you spot your strip, you run your strip, you actually then get this graph immediately after development. So you have bismuth 213 and francium 221 that was in the mixture before, that is not directly given off by the actinium that is currently there. And then you have also actinium constantly giving off daughter radionuclides. So after you wait for the daughter nuclides with the half-lives of 45.6 minutes and a few seconds to decay, you have a graph that looks like this, that is a more accurate representation of where your actinium is on your ITLC strip. So here you can see that there was no actinium here present when the strip was run to give you off some new daughter radionuclides, whereas here is all the actinium currently in your mixture. Like I said, there is another video on the channel that you can have a look at. Radiolysis is important, like I said, and method should be able to identify radioactive impurities up to 1%. The group at Erasmus is doing a lot of validation on HPLC methods, and recently they have also been showing that they can accurately measure radiolysis in actinium radiopharmaceuticals. But the HPLC method basically is a collection of fractions. So it's a normal HPLC method of a column. You separate your radiopharmaceuticals and the byproducts, and then you catch it up in you sample 15 drops or even sometimes less or more. They are optimizing this as well. And then you leave these samples to equilibrate with the daughters, and then you measure it in in a gamma counter system, and then you can plot it over time and actually get a good HPLC method. Importantly, actinium-225 production has to be considered. When you have actinium-225 produced as a radionuclide source, it is not stable over a very long time. The shelf life can range from 1 to 2 weeks. This is great, but remember the half-life of actinium is 10 days, and progressively while you label, your quality is going down. So you can imagine if you sit in Germany and you make your actinium-225 and you ship it to South Africa, like what I experienced in the past, it is already a week old when it gets there. And then you have like a limited time, like a week or a few days to radio label and dissolve this actinium and then use it to produce your radiopharmaceutical. Once you've produced your radiopharmaceutical, it again has a shelf life. And this is still under debate and being determined. And I really strongly believe if you want to prove a shelf life, you have to do a radio HPLC method. And then there is a time where your radiopharmaceutical is still stable and to be injected into your patient. It is always the question centralized or decentralized production, and this is therefore the things that we have to consider. I found this on LinkedIn where it was said that it was produced with a 5-day shelf life. I really doubt that any radiopharmaceutical currently with actinium has been validated to have a 5-day shelf life through a proper HPLC investigation, taking into account all of these stability issues, the recoil, the chemical release of the daughters, and radiolysis. Those are the three things that you have to measure, and through HPLC as well as ITLC, stuff like recovery, all of those things is important on your systems, and you have to validate also in the shipment conditions in your final formulation that you have a stable product. There is also an issue with the measurement of actinium-225 in preclinical work. The imaging methods are still being developed. You also have to wait when you take samples more than a day, 24 hours if you can, before you measure. You work in really low quantity, so it's also very difficult to accurately predict the dose that you inject into your animals. So yeah, if you want to pop a comment, if you've been working with actinium-225 preclinically, feel welcome. I'm interested to hear more. I've not done this myself. Then finally, actinium-225 dotatate is in phase 3 clinical trials. This is the most advanced application, and it looks quite promising indeed. Here is a bibliography. Have a look at that. Astatine-211. Again, pop a comment if you've worked with it. I've always been an admirer from afar because astatine-211 is quite complex and I always read up on it and hear what other people say about it, but I've not worked with it myself. My scope of expertise currently mainly lies in metal radio labeling, but it is quite an interesting isotope indeed. It is the heaviest element in group 17, and there is still an argument depending on which group you ask. I mean, not an argument in nuclear medicine or radiopharmacy or chemistry per se, but chemists all over other spectra of the chemistry society is still arguing if it's a halogen or a metal. Because it lies here in the metalloid range where the activities are predicted based on where it lies on the periodic table. Also important to note that astatine-211 is a very rare metal. Halogen, metalloid, whatever. A very rare element, and it has not been produced in high enough quantities that it's measurable in grams or stuff like that. So the most of the chemical analysis that is normally done is not feasible with the amount of astatine that we have. The best way to produce astatine-211 is through the bombardment of bismuth-209, natural bismuth, with alpha, and then the release of 2 neutrons gives you astatine-211, but you have to optimize the irradiation conditions to keep the amount of long-lived astatine-210 to a minimum. Most optimally 29 MeV or 28.5 MeV seems to be the way to go. There is a few cyclotrons in the world that are able to do this. Currently, some references say 37, others says 30. It's quite unclear how many cyclotrons, but 13 of the 37 is producing astatine at least sometimes. So there seems to be quite a great interest in the production of this radionuclide. And again, we have the problem of centralized production. The longer your astatine solution is being transported or shipped, the more the oxidation state changes and you have some radiolysis of the radionuclide, and there is therefore quite a shelf life on your astatine-211. The state your astatine-211 is and the time that elapsed after production can very well influence your radio labeling behavior. After you've labeled, there is quite a big question of the stability of astatine 211 radiopharmaceuticals, as we will go into that. Since astatine 211 gives one alpha emission, it's not linked to anything like recoil, but it's rather linked to chemical stability because of the large element that we are working with here. And therefore, I guess in a way you could say producing in a cyclotron and treating patients right next to the cyclotron in the clinic and having like a centralized cyclotron production of astatine 211 radiopharmaceuticals might be the way to go. The chemistry of astatine 211 is incredibly complex, and the oxidation states are assumed, like I said before, because we don't have a large amount of the element to do some investigations with. For organic incorporation in medicinal applications, halogen-like behavior is normally assumed, although some groups are kind of moving towards investigating the metalloid behavior of astatine 211. The pH in aqueous media and the presence of oxygenated species is determining the oxidation state for astatine 211. So astatine plus and astatine minus are the oxidation states that is used chemically, and the astatine plus is used for both halogen-like behavior or metalloid-like behavior, and astatine minus is used for halogen-like behavior. We have nucleophilic and electrophilic approaches. I'm not going to go into depth in this lecture on these, but there is also quite good references to go and have a look at. But it's important to know the electron affinity of astatine is lower than predicted or even lower than other halogens. So the amount of electrons in a bond that astatine 211 attracts to itself is a lot lower than expected, and this is also a very large element. The size of the atom is large, and the electrons spin in a quite a faster way than the corresponding element that it reacts to. So this leads to weaker halogen bonds than what is normal for the iodinated counterparts. Now, iodine chemistry forms the basis mostly of astatine chemistry, and a lot of the behavior that we want to predict for astatine 211 comes from iodine. And as we know, iodine is used quite widely in radiopharmaceutical production. So now we do deal with molecules that behave similarly, but the stability might be quite different. It was also proven in vivo that the metabolism of radioiodinated and astatinated compounds is different. So although this is feasible approaches to pursue, we should be clear that there is some unknowns, and this does influence really how much of the assumptions can we draw from iodinated counterparts. Astatine labeling of biological vectors use SAB or boron cages. You can read more about that in the review there. And then finally, astatine 211 applications that is the most advanced is the use of a astatine MABG, which is similar to MIBG of iodine. The use of astatine for thyroid cancer based on the mechanism that iodine is also taken up into the thyroid through the sodium supporters. And then also the radio-labeled antibodies and for leukemia. If you've worked with bismuth, type down below. I have worked with bismuth 213 now a little bit. It's quite an interesting isotope, perhaps viewed by many as not so useful. It has a short half-life of 45.6 minutes. It has a nice measurement 404 keV photon that you can measure, and it's used in metal-like chemistry with bismuth-free, the most prevalent oxidation state, and the production is dependent on the availability of actinium-225, unfortunately. Somehow, the general focus seems to be on the use of the in vivo generators rather than the alpha-emitting daughter. So actinium-225 has largely replaced the use of bismuth-213, and lead-212 has largely replaced the use of bismuth-212. So as discussed, the various production routes of actinium-225 will also have an influence on the availability of bismuth-213, and in this it is like the most hampering part of bismuth-213 availability. All the currently produced actinium-225 is being used in the various preclinical and clinical investigations out there, and you need a really high amount of bismuth-213 produced by the actinium-225 to get a clinically useful dose. So if you might know, actinium-225 is administered in doses from 6 megabecquerel up to 20 megabecquerel for radiopharmaceuticals. But for bismuth, you need more traditional amounts like 5 to 10 gigabecquerel should you be injecting it for systemic treatment. Now you need that amount of actinium-225 to give you that amount of bismuth-213, with actinium-225 having a yield of up to 80% of bismuth-213. So this is the generator system. It works very well. You load your actinium-225 on and you elute your bismuth-213 as it forms. Normally you can elute 2 to 3 times a day and it lasts you up to 3 weeks. There is the possibility to use the contaminated actinium-225 or 227 for loading up on the cartridge and then eluting it. This will lead to some waste because, of course, the actinium-227 is a long-lived isotope that you might have to send back. But what is nice is that these columns will capture both and you can just get out the useful bismuth-213. So it might be a way to upcycle or reuse actinium-225 batches that is not user-friendly for the clinic or patient use because of actinium-227 contamination. For bismuth-213, we have to be sure that we choose a chelator that very quickly labels, that is stable in vivo because free bismuth-213 goes to the kidneys and irradiate. It's really accumulating in the kidneys. It should be labeled at mild labeling conditions to allow the incorporation in biological molecules if needed. And then it should be labeling in high molar activity. So bismuth's distribution of complexes in aqueous solution is actually quite scary to see. So basically no pH is great for bismuth-213 and you can therefore label at any pH. Now this is really true. When you work in the lab, you can label bismuth in any pH, it will label. However, it does label quite easily. This graph looks like it will not label very well, but it is really mild and nice to work with. That being said, optimal chelator, more optimal than data, will allow you to label very quickly at mild temperatures because, as you can imagine, the 45-minute half-life of bismuth is quite detrimental. You have to label and do QC in as short time period as possible, and many new chelators are being developed. DTPA derivatives, NITA derivatives, DOTA derivatives, and azacron derivatives. It is also important that when you do preclinical work, the stability of bismuth complexes in plasma and serum is the most efficient way to predict in vivo stability. Applications include PSMA, but it was shown that actinium PSMA has a better dosimetry profile. The directly administration of bismuth peptides into the brain. It was also shown that actinium is not doing too bad compared. And then also for bladder cancer, monoclonal antibody that was injected directly. Direct injection into the lesion allows for lower dosages and also the use of biological molecules that will compensate for the short half-life of bismuth 213. Now we go to lead 212. Have you worked with lead? I have seen lead being labeled. I have not worked with lead, but it's quite interesting. So pop me a comment again below. It has nicely a stable isotope, which is quite rare or even not mostly possible for alpha emitters. I think terbium is the other one that has a stable isotope. So this makes it great for chemical characterization. You can really work on your cold compounds, do proper HPLC validation, and all of the other things that normally is quite tricky with alpha emitters. You also have the possibility of lead 213 for a theranostic pair for SPECT imaging. Keep in mind the infrastructure required to produce lead 213 will add a really high cost into the clinical realm for application. So currently it is perceived that lead 213 SPECT imaging will only be used for development purposes preclinically and maybe first in human, but that we will still use traditional other metal-based radionuclides like gallium and fluorine-18 for the day-to-day theranostic applications. It's an in vivo generator for bismuth 212. Be aware that the daughter thallium-208 has a high gamma emission, so if you're working with it in the radiopharmacy, you will have to have proper shielding, also during administration of patients, and patients might have to be kept for longer periods of time and isolated before they can be sent home. According to Richard Zimmerman in this article, more than 15 companies are working on offering lead to the market. So you can produce lead from various mechanisms: thorium-228, radium-224, radium-224, radon-220, and there is many generator systems being developed. So either you can use the parent isotope and just load all the mixture of isotopes and elute bismuth-212 out of that, and then recirculate it from time and time again and elute lead all the time. There is the radon gas generator that was published in 2023. It's quite a cool generator system, one I had the pleasure to see, and it's really quite interesting how it's being produced. You have the radon gas that is given off, then you leave it so that it decays on the glass vial, and then you wash the lead to 12 from there. And then there's also the production through photon conversion. During the process used to produce actinium-225, there is also the co-production of lead to 12, and this can then be isolated and be put to useful use. There is some progress on making hybrid chelators that can capture both other radiometals like gallium and so forth, as well as this lead with the soft chelation characteristics and a 2+ oxidation state, so that you can have one molecule to treat with lead, but also image with gallium-68, for instance. Then the most advanced in the clinic currently is dotamtate, and there is a phase 2 trial registered or coming up. Then going further, radium-223. If you are busy on nanoparticle development of radium-223 or targeted, or even just looking at the radiobiology of radium-223, please pop me a comment. It has an 11.4-day half-life, and it has the same characteristics as calcium due to being on the same level on the periodic table. So it's incorporated during bone formation in ion exchange, and of course in areas of high metabolic activity, incorporation increases. But we want it targeted, and this is quite tricky. So currently nanotechnology is being used. They are in various stages of investigation. No clinical investigations is registered yet. You have targeted, non-targeted, and then also just in-target retention, and yeah, they are also functionalized with various targeting moieties like PSMA or trastuzumab. A lot has been done in this, but the clinical translation, of course, is quite difficult, and there is a lot of hurdles, robust GMP batches being produced, toxicology, biocompatibility, regulatory uncertainty, and also the cost-benefit ratio of production. Some efforts is being made to incorporate rating 2 to 3 radium into the crown kind of chelators like Macropa. And then the clinical aspects are mostly still radium-223 as a calcium mimetic, along with some various forms of combination therapy. Terbium-149 is quite an interesting isotope. Maybe you have had experience with other terbium isotopes, so you can also mention that if you want. It has the absence of alpha emitting daughters, and you can image it. I mean, what better thing that we want? PET imaging of an alpha emitter. It has a very short 4.1 hour half-life, and yeah, it's extremely challenging production. And you can see there is a whole review down here. And of course, the people at the Paul Scherrer Institute has quite a lot of done quite a lot of research, and they were the group that now currently produced the most terbium-149 in one single production, 260 megabecquerel at 99% purity. I'm not going to go in depth into all of these mechanisms. I feel it's out of scope for this presentation and quite highly technical, but pop me a question if you have, and I will see what I do about that. It is a hard Lewis acid, and wow, it labels perfectly with DOTA, so there's even no need for optimization of the chelation, unless you want to do biological molecules, of course, because as we all know, DOTA labels at higher temperatures, but the radiochemistry is normally identical to that of lutetium-177, so all the optimization of lutetium-177 can be applied to terbium. So we are actually quite advanced with the chemistry, should be able to solve the production issues. The most advanced is currently published also by PSI in preclinical work. It's quite interesting to see. And there is a lot of publications also listed here by PSI, by Christina Miller, that you should go and have a read. Then finally, thorium-227. It has a long physical half-life, obtained from an actinium-227 generator, and you have a release of daughter radium-223 from chelator systems. Normally, because it has such a long half-life, the focus is on biological molecules. Currently, all of the research was being done by Bayer, and it's quite difficult to get hold of the details, but just some comments on thorium-227. There is a difference between the parent and daughter oxidation states of actinium-227 and radium-223, as you can see, +3 and +2, while thorium-227 really likes the +4 oxidation state, so you can easily remove it in a generator system. Should your thorium-227 be shipped far away and stand maybe 2 or 3 days or 4 days, you should be removing radium-223 again if your eluate is aged. DOTA is inefficient and definitely not for the mild temperatures required for the biological application that we want, because like I said, it's a long half-life, so you ideally want to match your physical half-life with your biological half-life. So we have to look at other ways to label biological molecules with thorium-227. And here are some examples that is being investigated is DFO, cyclostar, and HOPO. And it's important that an analogy between thorium-227 and zirconium-89 is being made. So all of the research going forward on zirconium-89 can also be translated or investigated for thorium-227. This is a summary of the clinical trials, so it is definitely still being investigated, mostly by Bayer. We will see what we can learn about this radionuclide in the future. So, my conclusion or confusion. I think This is a theory in progress and only my opinion, but basically what I want to say here is that we cannot evaluate a radionuclide in a bubble. It is multiple aspects that we have to look at, and things like clinical progression and upscalability always gets a lot of airtime, but we also should look at optimization of the chelation, the theranostic partnership, and is there recoil? So actinium, of course, the recoil is quite problematic in a sense, but it's also not the only radionuclide that has recoil issues. And I recently realized that this is some aspect that I still have to read up on, because really what recoil contributes to the clinical scene is still, for me, unclear. But what I can say is the stability of actinium-225 radiopharmaceuticals really needs a lot of optimization. Astatine 211, the chemical stability is something that we really have to fix. I put upscalability quite low here, but I think there is the benefit that if some infrastructure is being implemented, we will see this change in the future. Bismuth 213, I think the most problem here is upscalability due to the reliance on actinium-225, and then also the fact that you cannot centralize produce it. So bismuth 213 will always be from generator systems in small clinics, and this is possibly not so attractive for the pharmaceutical industry. Lead-212 looks overall quite promising, although I had a comment when I showed this at the conference that the recoil is possibly not so mild, and that I will have to read up on that too. So I will have a read on lead-212 recoil, and also then yeah, all of those quality control measurements and things that needs to be done. Radium-223 is not targeted, so this schematic is basically on the radium chloride, but the targeting should be fixed if we want to use it successfully for targeted alpha therapy. Terbium-149, I think the centralized production, upscalability, and clinical progression is all things that hamper that. So definitely upscalability of the production is urgently needed. And then thorium-227, of course, its clinical progression is unclear. There is quite a lot of daughter radionuclides in its decay chain, and also maybe the lack of a theranostic partner, although zirconium-89 could be utilized. And all in all, they all make a team. So all of the alpha emitters have strengths and disadvantages, and I think together they are strong. You can see together all of them cover all the needs, and based on what your facility prioritize, you should choose your alpha emitter to pursue. So the future of alpha emitters is unclear, but it's very promising and interesting and exciting, and we will keep on having all of these discussions. And depending on your outlook in life, you might like them or not. You might think they will change everything and we will only be having alpha emitters in the future. You might have your favorite alpha emitter, or you might really not like them and feel uncomfortable with them and the chemistry and the ambiguity. And of course, there's also radiobiology research to be done to figure out how they work exactly. So it's quite interesting, and I think we will have many more of these discussions. And since this was a conference presentation, I should actually still thank everybody, my amazing colleagues at work, the heads of the lab, Frederick Clearen and Guy Bormans, all the colleagues I've had coffees with in the past to figure out how alpha emitters work. And then also Prof. Adriano Duarte, which is a big mentor of mine and always is open for scientific discussions. The images drawn in this presentation is on a licensed version of PyRender. I have the license, so if needed, I can provide that. And you are already on my YouTube channel. Please like and subscribe and comment and interact with me. I have many requests for videos to come, but I'm also quite busy being a postdoc, so I have it on a list if you want to add some more ideas on the list for future videos, please do. And then I have to thank the Belgium Founders Research Foundation for actually sponsoring me as a postdoc researcher. So thank you so much. And yeah, I'm looking forward to interactions and your comments.